So my lab really interested in the human germline cell development, and we're also trying to develop some new technology. 14 years ago, when I still did postdoc research in Cambridge, so under the guidance of Professor Azim Surani and Dr. Kai Jin Lao, so we developed the first single-cell transcriptome sequencing technology, and seems the method really helpful for the developmental biology field and many other biological fields. And then in 2010, I came back to Sunny's Biopic Center. At that time, still there's no lab space, so I think Yan Yi spent 4 months to build up a new building, just about 1,000 square meters, and fully functional just within 4 months, then permit us to do the further research in Peking University. And then 3 years later, in 2013, so my lab developed the first single-cell DNA methylome sequencing technology, the single-cell RBS. And we realized that, in fact, it's a real single-cell whole genome methylation sequencing technology, because although it is based on the MSP1 cutting, but about 50% of the reads is from the randomly fragmented genomic DNA fragments. So if you're sequencing multiple single cells, you can cover the full genome accurately. And then we collaborated with Sunny, with Professor Jie Qiao, together on the human oocytes, the human germline development, both the pre-implantation development and the embryonic stage germ cell development. So with our work, so we find a lot of new exciting features of the DNA methylome reprogramming in our germline cells. For example, previously, people just assume during the pre-implantation, it's just the global demethylation. But we saw that, in fact, between the 4 and 8 cell states, there is also strong remethylation of the genome. And also, previously, people just assume the asymmetric demethylation of the paternal and the maternal genome is just removed and become a symmetric methylation. But what we found is, in fact, it is reversed. The hypermethylation on the paternal genome, and after 2 cell states, it becomes the hypermethylation of the maternal genome. And this even maintained to the post-implantation stage. And for the embryonic stage germ cell development, we also see a lot of exciting new features of our methylome reprogramming. And now we're also curious, what's the difference between the 2 waves of the eraser? Why the first round is just partially remove the methylation, whereas the second round is nearly all the methylation is removed. And then we also analyze other epigenetic layers, such as chromatin accessibility. And we find that, in fact, the chromatin accessibility or the open chromatin state is also reversed during the pre-implantation development in the human embryos, but is with the opposite direction. And we saw that this is a human-specific phenomenon, because in mouse, it is just the asymmetric chromatin state become symmetric and no reverse at all. And then we also analyze peri-implantation states, because we know at this stage it's nearly impossible to get the abortive embryos. So we're trying to using the in vitro culture system developed by 2 groups from UK and USA, and to do the single-cell multi-omics sequencing. And we see really how the X chromosome inactivation and the doses compensation happened in these peri-implantation embryos. And for the embryonic stage germ cells, we also see very strong unsynchronization of the germ cell development, much stronger than the mouse model. So this is the first time now we can see how during the development, how the male and the female embryonic stage germ cells just develop step by step in an unsynchronized way, and all the major features for every developmental point. And then I think after we do so many work, there's still possible maybe a lot of things we cannot using the current technology to resolve. And we find, in fact, there's quite a lot of such things. So we think we need to further improve our single-cell omics sequencing technology. So 10 years ago, Peter Fraser lab developed the first single-cell-Hi-C technology that really permit, no matter during development or any other in vivo situation, you can see the 3D genome structure in every type of cells at single-cell resolution. It's really powerful, but it's based on the NGS platform. So that means the real length is just 300 base pairs. So if you have one enhancer binding to one promoter, it's totally fine by using Peter's strategy to detect it. But if you have multiple enhancers, for example, you have 5 enhancers simultaneously binding to the same promoter, then the Peter's technology can only detect one of them, missing all the others. So now there is a single molecule sequencing platform with a long read. So we ask, can we further improve the method, permit us to see all these multiple interactions of the chromatin structures? And we saw that, in fact, it's possible. So we developed a single molecule sequencing platform-based single-cell Hi-C technology. We called single-cell nano-Hi-C. So basically, no matter it's just one enhancer binding to one promoter or multiple enhancers binding to one promoter, so we can sequencing the full fragment of this ligated product. Then we can see all of them. And we're also trying to optimize bioinformatic analysis pipeline because we know in general single-cell data is sparse and the coverage is relatively low. So we put a lot of efforts by Jensen to make it work. And then we saw that after you pulling about 100 to 200 single cells nano-Hi-C data together and compare to the bulk NGS platform-based Hi-C data, you can see we can capture the chromosome territories, the tag domains, and with very consistent with the bulk NGS data. So meaning the method really work quite consistently. And then we can also really capture these looping of the chromatin, no matter between enhancer promoter or other things. Here is our data, and here is the bulk Hi-C data based on the NGS platform. And then we know that majority of the cells in our body is the diploid cells. So it has 2 sets of the chromosomes. So Sunny's deep-seq method based on the NGS platform is already very nicely to separate all the 46 chromosomes within a single nucleus of our cells. But the problem is it based on the heterozygous SNP linkage. So if you use the NGS platform, only a relatively low percentage of the reads can be separate the paternal and the maternal allele. So now we're sequencing the full length of the Hi-C product. So we have a better coverage for these heterozygous SNP linked reads. So in this way, we don't need to sequence a single cell very deep, just about 5 gigabase state for per single cell. We can already separate all the 46 chromosomes in an individual nucleus. And this will be especially helpful for the X chromosome analysis because we know in the female cell, the X chromosome is randomly inactivated. So in this way, we can see, for example, in this GM cell line, the 2 X chromosome, the maternal one is loose, active, whereas the paternal one is more compact and silenced. So in this way, for every individual cell, now we can see how the 3D genome structure for each X chromosome and other chromosomes. And more importantly, if you use the NGS platform-based Hi-C analysis, then for the very complex structure variations, it's quite difficult to trace how it affects the 3D genome structure. But our long-read method can better resolve this issue. For example, in this K562 cell line, there's a very famous BCR-ABL1 fusion gene. So now within our method, we can clearly see at this fusion point around it how the genome's 3D structure is there. And how about the high-order interactions? And we see, in fact, we can really clearly identify this. For example, you can see this GM cell line we can detect around this important master transfer factor. You can see one promoter simultaneously binding to multiple enhancers, 2, 3, or even 4 or 5 enhancers. So that means it's very likely there's a synchronization of these different promoters in the same individual cell, simultaneously binding to the same promoter to regulate its transcription. And if you're looking at another gene locus, a very important long non-coding RNA is similar. One promoter is simultaneously binding by multiple enhancers in the same individual cell at the same time point. And then we ask if there's high order interaction between these enhancers and promoters. So do they really functionally important for the gene transcription? And then we just separate the genes in this cell line into the lowly expressed and highly expressed, and then see these high order interaction. And you can see these high order interaction enriched much stronger in these highly expressed gene compared to the lowly expressed gene. And it also enriched mainly in the A compartment, but depleted from the B compartment. So that means it's really these high order interaction very likely contribute to the gene transcription. So if you have a higher order interaction, then probably the gene expression is higher or more persistent. And then another type of the very complex structure variation is X chromosome circular DNA. It is very important for the cancer tumor genesis process because for the ecDNAs, it's very easy to accumulate a lot of copies compared to its linear chromosomes. And we saw that in this colon cancer cell line, there's a very famous circular DNA containing the CMYC gene. And it contains 4 different parts from 4 different chromosomes. And we saw that, in fact, for this ecDNA, within this molecule, there's a lot of looping permitting the enhancer and promoter to bind together to regulate MYC expression. And more importantly, we can even see this ecDNA binding to different linear chromosomes at other loci, probably offer the enhancer to the promoter on these linear chromosomes to activate the genes on the linear chromosomes. So that means now within this long-read-based single-cell Hi-C, we can really detect not just the complex structural variations, but how the epigenetic state changes on these new gene locus during the disease processes. And then we realize that, in fact, because the Hi-C data contains the long-read interaction information within the chromosomes, so maybe it will also helpful for the de novo assembling of our genome. And several years ago, we developed a long-read single molecule sequencing platform-based single-cell genome sequencing technology. And we saw that with just the single-cell genome sequencing data from about 30 individual cells, we can already assemble our genome into the contigs with the NG50 more than 1 megabase long. So that means more than 50% of our genome is within the contigs with at least more than 1 megabase length. And now if we pull in another, for example, 12 single cells nano-Hi-C data, we saw that we can even further assemble these contigs into scaffold with the NG50 of the scaffold more than 4 megabase. So that means, I think in the future, with the better coverage of single-cell genome sequencing data and the single-cell nano-Hi-C data, probably we can, using less than 10 individual cells, we can already assemble our genome into very long contigs and scaffolds. 6 years ago, my lab developed a single-cell multi-omics sequencing technology. Because so we applied our single-cell nano-Hi-C data also to the mouse embryonic states germ cell development. We're focusing on these 13.5 to 18.5 for both male and female. And you can see purely based on the single cell nano-Hi-C date, we can clearly separate different type of cells in these, for these germ cells, no matter is in mitosis or meiosis. And more importantly, now we can capture really in the male germ cells, the different cell cycle states, how the 3D genome structure of these germ cells. And for the female one, it contains both the mitotic one and the meiosis one. And we saw that for both the mitotic female germ cells and the meiotic female germ cells, for every critical cell cycle state, we can detect the 3D structure of the genome. So now we can really have a chance for these in vivo situation in our germ cells, how the 3D genome structure there and how it contributes to the gene regulation. Again, we find that for some very important master gene, we can see multiple enhancers simultaneously binding to the same promoter within the same individual cell at the same time point. And so that means these master genes regulation, it must be very complex and quite tightly regulated. If you're looking at different developmental states, we can also see it. And 6 years ago, my lab developed a single-cell multi-omics sequencing technology. Why we want to develop this? Because we want to know, for example, the chromatin accessibility and the DNA methylation is always first the methylation is removed from the gene promoter, and then it becomes accessible of the chromatin state, or sometimes it can reverse. So if you don't do the single-cell multi-omics to simultaneously detect both layers, you cannot answer this question purely by the bioinformatic analysis. And we find that, in fact, our method can very nicely resolve this. But one drawback of our method that it's based on the NGS platform. So the read length is just 300 base pairs. But we know for the CpG islands, very important DNA methylation regulatory unit quite often is longer than the 300 base pairs. In fact, the median length of the CpG islands is about 600 base pairs. So recently, we're also trying to develop a single-cell multi-omics sequencing technology based on the NGS platform, based on the single molecule sequencing platform, the Oxford Nanopore sequencing platform. We call it single-cell nano-COOL-seq. It can simultaneously analyze the transcriptome, the genome copy number variation, the DNA methylome, and the chromatin methylation, chromatin accessibility simultaneously in the same individual cell. In this way, we saw that basically we can identify the full length of the CpG islands for several hundred of them within every individual cell. And if we're pulling several hundred cells together, we can cover majority of these CpG islands and the gene promoters at full length, just within individual reads. And when we compare our data, chromatin accessibility data, to other technology measured bulk data, you can see it's very consistent between them. And we can also show that the method can clearly identify for every individual cell if it's a euploid cell or if it's a aneuploid cell very accurately. And more importantly, now for these very complex structural variations, for the first time, we can see the epigenetic changes. Again, for this BCR-ABL1 fusion gene in this K562 cell line around the break point, you can see we can really now determine how the methylation changes as well as how the chromatin accessibility changes. So now we can not only detect these structural variation event, but also see how it affects the different layers of epigenome. And we applied our method to a in vitro cultured cell system to see how the 5-AJ treatment to do the global demethylation, then how about the response of these cells. And we saw that, in fact, there's very strong heterogeneity, even at the same endpoint, within different individual cells for both the DNA methylation and the chromatin accessibility. So now means we really have a chance to see, even after the same treatment, the different cells can have different response, and then this different response, how it finally affects the gene transcription and the cell function. And you can also see, we can see both the down-regulated genes, up-regulated genes, and also the transiently up-regulated genes. And more importantly, we applied our method to the mouse pre-implantation embryos, focusing on the blastocyst stage embryo. And we can see the transcriptome aspect of our single-cell scNanoCOOL-seq data can clearly determine the different type of the cells and the developmental lineage of all these cells. And more importantly, now for the different time point or for the same lineage of the cells between different time point, we can see how the DNA methylation changes, no matter on the gene body or the promoter region. And comparably, we can also determine the chromatin accessibility between different lineages of cells and between the different developmental point. And in this way, now we can, in individual cells, we can see how the asymmetric chromatin state or DNA methylation between the paternal and the maternal alleles. For example, for this imprinting gene, the ICR region, you can clearly see for the maternal copy is fully methylated, whereas for the paternal copy is fully unmethylated. And on the contrary, if you're looking at the chromatin accessibility, the maternal allele is fully silenced, whereas the paternal allele is fully open. So now we can really see this in vivo during the real developmental process. And more importantly, because we can separate the paternal allele and the maternal allele within a single cell, then now we can even see the strand asymmetry of the DNA methylation in one DNA double strand molecule. So this way we can see the passive demethylation pattern during development for the first time now. Now you can see, for example, for the late blastocyst stage embryo, so basically the asymmetric of the demethylation within the DNA double strand, in fact, is stronger in the 12 ectoderm lineage compared to the pluripotent epiblast cells. So that means the passive demethylation in the 12 ectoderm is stronger than the epiblast cells. This exactly matches to the demethylation expression patterns. So I think now with this new long-read sequencing platform-based single-cell genome omics sequencing technologies, so now we can really identify the dark matters in different layers of our epigenome. For example, we can see the high-order chromatin interactions, we can see the structure variations, and how it affects different layers of our epigenome. And finally, I want to thank all my collaborators for happy collaboration. Thank you for your attention.